Okay, so we are live and ready to go for our tutorial today. And this is going to be a very gentle introduction to principal component analysis in cytometry for dummies. So yeah, again, welcome to today's tutorial. My name is Ramji Srinivasan, and if you are interested in playing along at home, you can play around with the demo data set at app.takeo.bio. So again, today's tutorial is about principal component analysis in cytometry for dummies like me. And this is going to be a very gentle introduction to a very powerful tool. And just as a reminder, our mission at Takeo is to make high parameter or high dimensional cytometry simple and easy for clinical trials. So who might care about today's topic? It's anybody who wants to find the essential drivers in a big cytometry data set. And this tutorial is heavily inspired by StatQuest and several others, so please like and subscribe to StatQuest channel. They do amazing work. In short, what it means is if you have a big matrix that is, say, 33 dimensions, 33 cell populations times 24 subjects, you can use magic like principal components to reduce this 33-dimensional data set to 2 dimensions and capture a lot of the variance. This is a preview of what we're going to walk through, but this is an example study comparing young, middle-aged, and centenarians and seeing the different immune profiles for these different patients. So how do we get here, and what does that all mean? What we're going to do is step through a synthetic data set inspired by real-life Lugli et al. In 2007. So this was a experiment designed for this particular for this particular paper. They had 24 subjects, 9 young people, 8 middle-aged, 7 centenarians, and they captured 33 T-cell subsets from an 8-color flow panel. But you can imagine capturing hundreds, thousands, or millions, or potentially billions with unsupervised clustering. So we're going to show the synthetic data set, and which is inspired by this, but then we'll also show the real-life findings to compare those. So for example, for each patient, they had a blood specimen, they ran it through a cytometer, and then they captured 33 populations, which are the rows over here, and then the columns are the individual subjects. So for example, the individual young people over here and centenarians on the right. And you can see here the percentages, which are the percentage of the immune state captured by or of this particular cell type that this person had, like 2%, 3%, et cetera. So these rows and columns are what we're going to do principal component analysis on. So let's step back a second here. So we said 33 populations by 24 subjects. If we just do 2 dimensions, so we take a blood specimen, put it in a cytometer, and just capture 2 populations, and we have a lot of data, it's easy to plot 2 dimensions on an XY plane. So let's go back over here. So let's take this young one, 75% and 2%. We can plot somebody like this on an XY plane because we're only capturing 2 dimensions. So young one over here, young 2 over here, centenarians over here, etc. And let's also, reiterate that this tutorial is only useful if you are interested in caring about the differences between different populations. If you don't care about the differences between, say dose groups, response groups, then this probably isn't a useful tool right now. But if you do care about the differences, then principal component analysis is awesome. So let's bump it up to 1 more dimension. So went from 2 dimensions to 3 dimensions. So now we have individual cell populations over here, cell population 1, 2, 3, and the individual young people, and then centenarians on this side. At 3 dimensions, this is also not so bad. You can see the X, Y, and Z plane over here, and you can see the separation between centenarians and young people. So remember, in the X, Y, Z plane, we can plot all of these dimensions. And we can see this kind of clustering, and we can see this kind of separation. But what happens when we go to 4 dimensions, or 30 dimensions, or 298 dimensions? And this is where minds get blown. So the human brain can only visualize in 3 dimensions. And so this is where PCA, also known as principal component analysis, comes in. And there's going to be some fancy mathematical jargon, but we'll hopefully break it down. A reminder, PCA is a way to see the essential drivers in a big cytometry data set in 2 dimensions or 3 dimensions. So if you've got 40 dimensions or 100 dimensions or whatever, you want to reduce it down to 2 or 3 dimensions, but not lose a lot of the variation in the underlying data set. So this would be the end product. If you have a 33-dimensional data set, you'd ultimately be able to reduce it down to a 2-dimensional data set, as we showed in the beginning, with centenarians, middle-aged people, young people over here. You can see PC1 and PC2 are little variables here that we're going to show in more detail. And these have numbers here of the amount of variance in the data set they capture. So 24.18 over here and 12.23% variance. If you sum these up, the casual reader will note these constitute 36% of the variance. So we're going to walk through exactly how these are computed and what they mean for your data set analysis. So how do we get to this particular 2-dimensional plot? We started with this 33-dimensional plot with 33 populations in 24 subjects. And then we computed what's called a covariance between variables. So over here, this is an excerpt of a matrix where we have 5 individual cells. And for each individual cell, we compute how much it covaries with itself and how much it covaries with other different cells as well. So for example, CD4, TN, CD127, CD95 minus, CD38 dim with itself, and then with another variable, with another immune cell, et cetera, et cetera. And the diagonal will show either signal or noise. So a high value over here is probably interesting. A small value is probably noise. Off diagonal is redundancy. So large values are redundant and small values are going to be noise. And so we're going to walk through to build your intuition for that. So let's go down to a smaller version of this data set, or excuse me, a dummy version of this data set, and to explain that in more detail. So remember, on the diagonal of that covariance matrix, large values are interesting, and off diagonal, these are, the off diagonal shows redundancies, so large values are redundant and small values are noise. So these are the raw population percentages of T cell subjects across subjects, T cell subsets across subjects. So if you take a sample over here, like high variance T cell subset 1, this varies a lot as you look from left to right. So you go from subject 1 to subject 10, this is going up and down quite a bit. Same with this subject as well. This noise subset that we've labeled isn't changing that much. So as you go from left to right, it varies very little. Off diagonal, these look quite similar to one another. So if you look 19, 11, 10, et cetera, so T cell subset 4 and T cell subset 5 look very similar to one another. So they're likely going to be noise. And then off diagonal, these vary a lot from each other. So these are likely to be useful. So if you think about this high variance population is likely to have a high covariance with itself. This noise population is going to have a very low covariance with itself. It's not going to change that much. These redundant populations will have a low covariance with one another. And these useful populations are very likely to have a high covariance with one another. So this is very, going to come in handy in our covariance diagram later. So what principal components goal is to drop noisy populations and focus really on the signal populations and potentially eliminate redundant populations as well. So let's go then to more detail. So zooming out to all 33 populations. So here you have the covariance matrix, and high values on the diagonal are signal, and you can see basically high values on the off diagonal are going to be characterized by covariance between those two particular populations. Oops. So now let's zoom out to all 33 populations. So you can see over here, the diagonal, you'll see a lot of signal on these particular values. And then the off diagonal, you're going to see potential markers of redundancy. So if two variables vary together, then they have a low covariance. And if two variables are basically if two variables vary together, they have a low covariance. So this could be potentially interesting for us. So now we're going to compute the eigenvalues of this matrix. So eigenvalues are a very complex term, and we're not going to go into the entire derivation here. But from this big matrix, you get all the individual principal components here, and then individual eigenvalues. So eigenvalue 1 is 3024.8. That's 24% of the total variation. So this is computed by taking 3024 and summing this entire column together and getting to 24%. So this captures a lot of the variation in the population. Principal component 4, 573.7, this is 8.17%. So 573 divided by the entirety of this column gets you to 8.17%. So here's a definition here. The PC1 axis is the first principal direction along which the samples showed the largest variation. Let me pause again here. This is beyond the scope of this tutorial to explain eigenvalues and eigenvectors. There's a great tutorial linked here that you will be able to see in the YouTube, and that's from 3 Blue, 1 Brown. So if you're interested in that, feel free to check that out. But let's assume you have these principal components derived for your cytometry data set. So you've got PC1 through PC7, you've got all these individual eigenvalues. Each principal component is going to correspond to a set of loadings. So let's take PC1 and this particular eigenvalue, and you're going to go down over here and you can see these coefficients are going to tell you which subsets matter the most. So for each cellular population, you're going to see PC1 loadings, negative 0.237, 0.195, 0.183, 0.307. And we're going to be able to plot XY from these PC1 and PC2 loadings. So we start here with this raw percentage, 75.4%. And these coefficients will tell you which subsets matter the most. So PC1 loadings, PC2 loadings. And we multiply through. So 75.4 times negative 0.237 gets you to 17.870. 75.4 times 0.156 gets you to 11.725. So this is what we're going to plot on the XY plane. So this is the PC1 and PC2 axes, and you can see the centenarians, the middle-aged, and young people all plotted. So we reduced 33 dimensions down to 2 dimensions by taking PC1 and PC2. So what is the value of this entire exercise? We've captured a 33-dimensional data set in 2 dimensions. So you've got a big cytometry data sets with dozens, hundreds, or thousands of cells measured by specimens, and you want to see what drives the difference between an endpoint, like response or dose. Now, bonus is to explain correlation circle plots. This is a way to graphically represent the relationship between the original variables and the principal components. So for these individual T cell populations, you're going to have the position of the variable on the plot and the eigenvalues, which are going to describe the magnitude of variable contribution. So for example, PC1 loading of 0.29 is high, and then you've got 0.13 and 0.12. These contributions are quite low. So let's see how to read a correlation circle plot. So over here, you'll see similarly the PC1 and PC2 plots, and you'll see different cell populations. Let's take this one, CD4 positive, TN, CD127 positive, CD95 positive, CD3 dim. The magnitude, long arrows contribute more to the variation, and short arrows don't contribute that much. And if it's more horizontal, then it contributes more to PC1, and more vertical, it contributes more to PC2. So this is showing a correlation circle plot for the top 5 T cell population. So let's go to some real findings from Lugli et al. And this actually came from the real life data set. So you have young patients over here, middle-aged patients over here, centenarians, and their PC1s and PC2s and PC3s were much better than our synthetic. They had 60% variation in the first principal component, 28% over here, 4.77% here. So they captured a lot of the variance, like 87 plus almost like 91, 92% from all 3 components. So they were capturing a lot of the data set variation in their real life data set. And they found that young donors in advanced age were different based on memory T cells, and specifically these differentiation markers were up in young donors, and these differentiation markers were up in advanced age. So now let's turn to a TACO data set. So we offer PCA on app.taco.bio, and this came from University of Minnesota in 2022. This is a phase 2 clinical trial with 40 subjects, 20 specimens, 20 responders, and 20 non-responders. So let's click on over to see this data set, which you can also play around with. All the credentials are over here, so you can log in whenever you like. We're going to go to the UMN data set here, and we're just going to quickly reorient ourselves. Remember, there were 40 samples, 20 subjects over here. And let's go to group similarity. And here you can see the principal component analysis and the explained variance, PC1, 55%, PC2, 19.6. So these do a good job of capturing the variance. So let's color by response so we can see a little bit of separation between responders and non-responders. And then further down, we're going to, let's shrink this down to 7 contributors so we can see different contributors. What is driving the difference? So for example, classical monocytes are driving a big difference. CD4 positive T central memory are driving the second most. And cytolytic NK cells, which one wouldn't necessarily expect on a classical flow panel, are the third here. So yeah, you can see that in a little bit more detail over here. So classical monocytes all the way down to cytokine producing NK cells. So this could tell you what drives the difference in treatment response as well. So wrapping up, PCA is particularly useful when the data set is highly correlated. So you can assume that there may be redundancy in the data, or that you can potentially drop variables without losing an explanatory power. So this is a great quote, which is PCA can be used to reduce original variables into a smaller number of new variables, but still explain a lot of the variance of the original variables. In fact, you can even quantify how much is retained afterwards. So with that, we'll wrap up. And if you're interested in more, you can check out app.taco.bio. We have tons of data sets there that you can play with. And if you've got any further questions, feel free to email us or contact us at taco.bio. So thanks for signing.